Good evening, everybody. Welcome back to regular daily lectures from New Age Cosmetic Biotech Healthcare Consultancy. We continue with the subject of molecular biotechnology at chapter number 5, which deals with bioinformatics, genomes, and proteomics. We started and are continuing with the subject of serial analysis of gene expression. Unlike DNA microarrays that rely on hybridization and signal detection, SAGE uses recombinant DNA techniques to clone randomly linked short sequences of cDNA prepared from extracted cellular mRNA that can be efficiently sequenced to identify expressed genes, polyadenylated mRNA, and was captured by an oligo-DT sequence that is labeled with biotin and attached to a strep-to-bead encoded magnetic bead. I'm repeating the last finished portions for the sake of continuity, and then we go on to the next part of the same subject, right? Double-stranded cDNA, excuse me, is synthesized from the purified mRNA using reverse transcriptase to synthesize the first strand of cDNA from the oligo-DT primer, an mRNA template, and then DNA polymerase to synthesize the second complementary strand. A strong magnet is used to retain the magnetic beads with attached cDNAs. During successive treatments and washings, the cDNA is cut with restriction endonuclease NIA3, which results and recognizes the sequence and cuts outside the GC base pairs, leaving a 3' GTAC extension. Since NIA3 cuts on average 1 in 256 base pairs, BP, there is a high probability that each cDNA will have at least one NIA3 recognition site, and because the cDNAs are bound to beads, each NIA3 cut site that is closest to the 3' end of cDNA is retained, and all unbound fragments are washed away. In the nomenclature developed for SAGE, NIA3 is called as anchoring enzyme. Next, the NIA3 digested cDNA sample is divided in two. One aliquot is ligated with adapter A and the other with adapter B. Both adapters have a CATG extension that is complementary to the extension produced by NIA3 digestion. A 5 base pair recognition site for the restriction endonuclease BSMF1 and a sequence of 4' in a PCR. The adapters have different primer sequences to prevent intra-strand base pairing snapback during subsequent PCR steps. After the adapters are ligated to NIA3 digested cDNA, the products are treated with BSMF1. Unlike NIA3 and other type 2 restriction endonucleases, this type 2's restriction endonuclease cuts 10 nucleotides downstream from its recognition site in one DNA strand and 40 nucleotides in the other strand, regardless of the intervening nucleotide sequence. In SAGE, BSMF1 is known as a tagging enzyme, and the segment of cDNA produced by the BSMF1 treatment is called a tag. BSMF1 digestion releases the adapter tag molecules from the beads into solution, from which they can be recovered. The 4 nucleotide extension of the BSMF1 cut DNA is filled in to form a blunt-end molecule. The pools of adapter A and adapter B tags are mixed. The T4 DNA ligase is added to the mixture. Under these conditions, the blunt ends of 2 tags are joined to form a 2-tag molecule, a D tag. That is flanked by primer sequences. Since the tag formation is completely random, tags from different cDNAs are joined during ligation, and the D tags are readily amplified during PCR using primer sequences present in the adapters. The amplified D tags are treated with NIA3 to release the adapter sequences and produce D tags with an NIA3 extension at each end. The NIA3 D tags Digested tags are ligated to form multiple randomly joined combinations, concatenamers of the tags. Concatenamers that are of about 500 pp in length are isolated and cloned in an E. coli plasmid. The concatenamers are sequenced. The sequence of each tag is recorded, and a specialized tag to gene database is searched to identify the corresponding gene. And the sequence tag is derived from the 3' end of the mRNA and therefore corresponds to the 3' end of the gene. The number of times each tag is sequenced, which represents its abundance in the initial sample, is determined. Up and down regulated mRNAs can be identified by comparing the frequencies of tags in different samples. Generally, more than 10,000 unique tags are collected from a single experiment. Over 30 million SAGE tags have been assembled from humans and another 35 million from various organisms. In principle, SAGE can detect all the transcripts in a sample, whereas with a DNA microarray, only the sequences that correspond to probes on the array are identified. Additional SAGE protocols such as long SAGE and super SAGE have been developed to produce longer tags, table 5.5. Also, other anchoring enzymes, example SAU3AI, and other restriction endonucleases that recognize and cleave at specific 4BP sequences, 4-base cutters, have been used to identify transcribed genes that do not have a convenient NIA3 site. An online resource called SAGE-NE and the ad here browser ad is available for matching tags to likely genes, determining the frequency of a tag among various SAGE libraries, and providing other pertinent information. The total RNA of poly A plus mRNA purification leads to cDNA synthesis, cut with NIA3, and leads to the split sample ligate adapters, adapter A, primer A, BSM1, F1, primer B, BSM F1, adapter B, and primer A, primer B, cut with BAM F1, primer A and primer B leads to the final product. And here, figure 5.9 explains the SAGE poly A plus mRNA transcripts are isolated by poly-DT hybridization. The poly-DT sequence is biotinylated B and bound to streptavidin SA-coded magnetic beads, yellow circles, or the poly-DT is attached directly to magnetic beads. Double-stranded cDNA synthesized from the captured mRNAs and then cut with restriction endonucleases and IA3. The fragments of cDNA that are not bound to the magnetic beads are eluted. The NIA3 cut cDNA sample that is attached to the magnetic beads is divided into two. One sample is ligated with adapter A and the other with adapter B. Each adapter contains a 4-base extension that is complementary to the 4-base extension produced by cleavage with NIA3, a recognition site for the restriction endonuclease BSM F1, and its specific primer sequence primer A and primer B. The ligated adapter NIA3 cut molecules are treated with BSM F1, which cleaves the DNA 10 and 40 nucleotides downstream from its recognition site, open arrows. The extensions of the BSM F1 fragments are filled in by DNA synthesis, and the mixture is blunt-end ligated. Semi-ligated molecules consist of two joint segments with different cDNA tags that are flanked by sequences for primers A and B. The D tags are amplified by PCR and then treated with NIA3 to remove the primer sequences and generate sticky ends. Ligation of NIA3 cuts D tags, forms concatenamers of various D tags. Concatenamers with about 20 D tags. Approximately 500 base pairs are purified and cloned into a plasmid vector. The concatenamers are sequenced and the individual tags are identified. The likely corresponding gene is determined by a similarity search, and the frequency of each tag is sampled in the sample is recorded. This is the fill-in tags of primer B and blunt and ligation D tag, primer B, BCR, cut with the NIA3. Please take our screenshots and make notes. Concatenamer formation. Going over to size separation of concatenamers, 500 base pairs, clone into plasmid and maintain in E. coli, DNA sequencing of tags, identification of tags, count the individual tags. And then you have the SAGE system of different tables 5.5, system, tag and enzyme, recognition site, tag BP. Now we're on to the subject of proteomics. Proteomics is the comprehensive study of all the proteins, that is the proteome of a cell, tissue, body fluid, or organism from a variety of perspectives, including structure, function, expression, profiling, and protein-protein interactions. There are a number of good reasons to study the protein complement of cells or tissues. First, proteins comprise the active components of cells. They are the molecular machines that catalyze the synthesis of important metabolites and molecules, monitor the internal and external environment of the cell, and mediate responses to environmental perturbations, and make up the structural components of cells. Thus, insights into the proteins that are present in a cell or tissue under particular biological conditions can aid in our understanding of the cell's activities. Although protein coding sequences can often be identified in genomic sequences, some are not annotated open reading frames, ORFs, or open reading frames are subsequently found not to encode proteins. Others encode proteins whose functions cannot be predicted from the sequence. Furthermore, post-translational modifications that influence the protein function and cellular localization of proteins often cannot be predicted from the sequence. On the other hand, a protein's function can sometimes be inferred by determining the conditions under which it is expressed and active, although expression profiles of protein shall be expressed after box 5.1. How many genes do we have? We proceed with the box 5.1. How many genes do we have? The exact number of genes in the human genome has been difficult to pin down. Before the release of the draft sequences of the human genome in February 2001, it was thought that we have between 80,000 and 100,000 genes. Surprisingly, estimates based on analysis of the draft sequences range from 23,000 to 49,000, with a consensus value of about 30,000 genes. By comparison, yeast, S. cerevisiae, fruit flies, T. melanogaster, and roundworm C. elegans have about 6,000, 13,600, and 20,000 genes, respectively. Considering our apparent biological complexity, it was perplexing that humans have only 50% more genes than a simple invertebrate and about as many as a tomato. To confound the gene number controversy, some researchers estimate that there were as many as 60,000, 89,000, or quite possibly 120,000 human genes. Clearly, a number of questions come to mind. How are gene numbers predicted? Why do gene counts vary? What is the likely number of the human genes? What is the significance of a low gene number? One of the obstacles to an accurate human gene count is that protein coding genes make up only 3 to 5% of the total genome. Although it is a bellabowed cliche, gene counters are looking for a handful of needles hidden in a large haystack. There must be a precise identification process for any realistic tally of the total number of human genes. Unfortunately, a eukaryote gene structure confounds gene enumeration. Exons are usually about 140 nucleotides and are separated by introns. There may be thousands of nucleotides in length. Also, the sequences at the 5' and 3' ends, 5' and 3' untranslated regions of mRNAs are not translated and vary from gene to gene. However, there are some consistent features that are in the identification of protein coding regions, ORFs, and that can be readily recognized. The translation start codon ATG distinguishes the first exon at the beginning of a protein coding gene and is preceded by transcription start site. At the 3' end of most genes, there are a translation stop codon, transcription termination site, and sequence that signals poly-A trait tailing. Finally, the splice sites that precede and follow the internal exons are regular motifs. A number of gene prediction computer programs based on the common gene structures have been devised to scan the genome for possible genes. Generally, these programs are designated ab initio, which means from the beginning or in the context of gene finding, an approach based on first principles. The ab initio programs predict coding nucleotides with high accuracy and recognize exons with good efficiency, but are less than 50% effective at finding complete genes. In addition, the number of falsely identified genes, false positives, are high. Alignments between a comprehensive set of cDNAs and the human genome are another way of identifying genes. Full-length cDNA, which are derived from extracted cellular mRNA, are an excellent resource. This aspect will be continued. The separation and identification of proteins. Ordinarily, the study of proteome of an entire multicellular organism is difficult due to the diverse functions of the many cells, tissues that comprise. I'll be back once we finish that sentence on the box. Now, coding sequences can be determined using transcriptomics. mRNA levels do not always correlate with protein levels, and interactions between proteins cannot be assessed by these methods. From a practical standpoint, proteomics can be used to track clinical disorders and detect targets for therapeutic treatments. A number of factors complicate the study of proteins. And first, in eukaryotes, there are many more proteins than genes due to alternative splicing, post-translation modifications, and to a lesser extent, post-transcriptional modifications to RNA editing, with about 60 to 70% of the estimated 30,000 human genomes, as shown in box 5.1. Undergoing alternative splicing, the human proteome may consist of 85,000 or more different proteins. Second, it is impossible to account experimentally for every member of proteome with a single technique because proteins are susceptible to degradation, have different properties, including different solubilities, and range considerably in abundance. In spite of these drawbacks, effective procedures have been devised for examining most of the components of many proteomes. Separation and identification of proteins. Ordinarily, the study of the proteome of an entire multicellular organism is difficult due to the diverse functions of the many cells and tissues that comprise the organism. Box 5.1 continued. For finding genes, because they contain all the exons of a gene, however, variation in the splicing of exons makes accurate gene identification somewhat arduous. More importantly, relatively few complete human cDNAs have been synthesized and sequenced. All the techniques have been developed for capturing intact capped mRNAs. The major sources of cDNA sequences are EST databases. These cDNAs are incomplete and concentrated at the 3' ends of the mRNAs. In addition, the databases contain about 10 to 15 ESTs for each gene. In some studies, a single representative sequence for each cDNA was determined to winnow down the overall number of cDNA sequences that would be needed for scanning genome sequences. The cDNA approach effectively locates exons. However, final gene counts are determined by combining the information about cDNA alignments with the results from ab initio analysis. A number of genes are overlooked with this approach because not all genes are represented in the cDNA databases. Also, miscounting splice variants as a number of individual genes instead of a single gene can inflate a gene count. Theoretically, the alignment of the complete genomic sequence of an organism, such as the mouse or pufferfish, to the entire human genome should locate highly conserved sequences that would likely be exons and regulatory elements and produce poor similarity scores for introns and the DNA between genes, intergenic DNA. The principle underlying this comparative genome strategy for gene enumeration is that introns and intergenic DNA are not under the same biological constraint as exons and regulatory regions. Thus, nucleotide changes accumulate in introns and intergenic DNA, with the result that over a long period of time, these sequences in different species diverge from one another. By contrast, the sequence similarity of exons and regulatory elements is maintained between relatively closely related organisms because gene mutations lower reproductive fitness, which makes these sequence deviations less likely to be passed on from one generation to the next. Generally, on the basis of different gene identification strategies, we probably have about 22,000 genes. The ENCODE, Encyclopedia of DNA Elements, project is a large collaborative effort that is dedicated to elaborating in fine detail the molecular aspects of the human genome. Some important and exciting discoveries have been made by the ENCODE pilot project, which covered about 1% of the human genome. For example, virtually all the DNA segments under study were transcribed. Many coding regions have multiple transcription start sites, and surprisingly, transcription often goes in both directions within the same segment of DNA. Most studies are required to get a broader view of how the human genome functions. However, not only does the ENCODE pilot study point to more human genes being discovered, but the apparent complexity that has been observed strongly suggests that it may be necessary to redefine the current concept of the gene at the molecular level. I repeat, it may be necessary to redefine the current concept of the gene at the molecular level. Now, for convenience, the complexity is often reduced by examining the protein complement of a cellular component or organelle, such as the nucleolus, nuclear matrix, lysosome, or endoplasmic reticulum. These protein subsets have been dubbed as subproteins because of its high resolving power. Two-dimensional polyacrylamide gel electrophoresis 2D page is frequently used to separate a population of proteins in a sample. Briefly, the proteins in a sample are first separated on the basis of their net charge by electrophoresis through an immobilized pH gradient in one dimension. The first dimension. Amino acids in a polypeptide have ionizable groups that contribute to the net charge of a protein. The degree of ionization protonation is influenced by the pH of the solution. In a gel to which an electric current is applied, proteins migrate through a pH gradient until they reach a specific pH, the isoelectric point, where the overall charge of the protein is zero and they no longer move. However, some proteins migrate to the same position in the pH gradient because they have the same net charge, although they have different molecular weights. These are further separated according to their molecular weights by electrophoresis at right angles. The first dimension. The second dimension, through a sodium dodecyl sulfate polyacrylamide gel, the separated proteins form an array of spots in the gel that is visualized by staining the spots with the Kumesi blue or silver protein stain. A 2D polyacrylamide gel can resolve up to 12,000 different proteins. The pattern of stain spots is captured by densitometric scanning. The pattern of stain spots is captured by densitometric scanning. Databases have been established with images of 2D polyacrylamide gels from different cell types. Software packages are available for detecting spots, matching patients between gels, and quantifying The protein content of spots. Proteins with either low or high molecular weights, those that are found in cellular members and those that are present in small amounts, are not readily resolved by 2D page. Also, highly charged proteins such as ribosomal proteins and histone proteins are not separated by standard conditions. The next task after separation of most of the proteins of a proteome or subproteome is to exercise the individual proteins from the gel, often using robotics to extract large numbers of proteins and to identify as many of the proteins as possible. Mass spectrometry, MS, is commonly used for this purpose. In principle, a mass spectrometer detects the mass of the ionized form of a molecule. For protein identification, proteins are fragmented into peptides that are ionized and separated according to their mass to charge, M by Z ratios, and then the abundance seen and M by Z ratios of the ions are measured. The results are presented as a spectrum with x-axis representing M by Z ratios and the y-axis representing the abundance of each ion relative to the most abundant ion. In practice, mass spectrometers have different configurations according to the nature, wet or dry, of the sample analyte, the mode of ionization of the analyte, how the electric field is established by accelerating the ions in order to separate and sort them, and the method for detecting the different masses. Mass spectrometric studies of proteins and peptides have been facilitated by effective ionization methods such as electrospray ionization (ESI) and matrix-assisted laser desorption ionization (MALDI). Peptide masses are usually determined by MALDI MS and amino acid sequences by ESI tandem. MS, ESI, MS, MS. MS is an important proteomic tool because that is mass spectrometry tool because analyses are rapid and accurate and require small amounts of starting material. Moreover, computational protocols are available for processing large amounts of MS data. Protein identification is straightforward because particular databases can be easily searched with either peptide mass or amino acid sequence data. For whole proteome analysis, the cellular proteins must first be separated, typically by excision of individual protein spots from a 2D polyacrylamide gel following electrophoresis. Each excised protein is then treated with the proteolytic enzyme trypsin, which cleaves on the C-terminal side of lysine and arginine residues. Contaminating salts, polyacrylamide, and other compounds are removed before the peptides are concentrated. MALDI MS is used regularly to determine the mass M by Z value of each peptide fragment generated from the excised protein. Briefly, peptides are ionized by mixing them with a matrix consisting of an organic acid and then using a laser to promote ionization. The ions are accelerated through a tube using a high voltage current, and the time required to reach the ion detector is determined by their molecular masses. With lower mass ions reaching the detector first. With lower mass ions reaching the detector first, the values of the observed peptide masses are matched with the expected masses of tryptic peptides for all known proteins. This type of analysis is called peptide mass fingerprinting. Online site example http://.xpc.ch/tools/al-dente/ are available that rapidly find the set of peptide masses of a known protein that most likely corresponds to those of the unknown proteins. In figure 5.10, which is just before the present explanation, the 2D page for separation of proteins is shown here. First dimension isoelectric focusing is performed to first separate the two proteins in a mixture on the basis of their net charge. The protein mixture is applied to a pH gradient gel. When an electric current is applied, proteins will migrate toward either the anode plus or cathode negative, depending on the net charge. As proteins move through the pH gradient, they will gain or lose protons until they reach a point in the gel where the net charge is zero. The pH in this position of the gel is known as the isoelectric point and its characteristic of a given protein. At that point, the protein no longer moves in the electric current. B. Second dimension. Several proteins in a sample may have the same isoelectric point and therefore migrate to the same position in the gel in the first dimension. Therefore, proteins are further separated on the basis of differences in their molecular weights, MW, by electrophoresis at a right angle to the first dimension through a sodium dodecyl sulfate polyacrylamide gel. This is an illustration of the pH and molecular weight where two-dimensional polyacrylamide gel is shown. And it shows a two-dimensional polyacrylamide gel and the excited spot, trypsin treatment, protein database, peptide mass determination, MALDI, TOF, MSH, and computer-derived tryptic peptides, peptide masses shown here. And we have the matchup peptide masses with explanation in figure 5.11. Now, peptide mass fingerprinting, a spot containing an unknown protein that was separated by 2D page is excised from the gel and treated with trypsin. Purified trypsin peptides are separated by MALDI time of flight, MS. The set of peptide masses from the unknown protein are used to search a database that contains the masses of tryptic peptides for every known sequence protein, and the best match is determined. The trypsin cleavage sites of known proteins are determined by the amino acid sequences, and consequently the masses of the tryptic peptides are easy to calculate. Only some of the tryptic peptide masses for the unknown protein are listed in the example. Now, alternatively, the amino acid sequence of a peptide can be obtained by ESI, MS, and MSH, and used to search a protein database to identify an unknown protein. With this approach, the peptides derived from a protein spot in a 2D acrylamide gel are first separated by mass, and then one of the peptides is selected for sequencing. Fragmentation and ionization of a peptide occur by cleavage along the peptide backbone. That's amide bonds, peptide bonds between amino acids. When the charge from ionization is retained at the N terminus, it is designated as a B ion. And when it is retained at the C terminus, it is a Y ion. Each ion type forms a ladder of subsequences that differ in size and consist of 1, a 2 dipeptide, 3 tripeptide, and more amino acids up to the full length peptide, an amino acid sequence. An amino acid sequence is determined from the mass values of ions of the same type, that is B ion or Y ion, by calculating the difference in mass, delta M, between subsequences. Thus, difference for members of a Y ion ladder represents the successive loss of an amino acid from the N terminus. For the Y ion subsequences B, E, we have D, E, F, K, F, D, E, F, K, D, E, F, K, E, F, K, F, K, and K. The difference from one subsequence to the rest is the removal of one amino acid from the N terminus. In other words, for amino acid sequencing, the Y ions form a reverse mass ladder. The converse holds true for a B ion spectrum. Each difference of mass is equivalent to the mass of a known amino acid, except for leucine and isoleucine, which have the same mass. For example, with part of Y ion series, the mass by charge ratios, M by Z values, for 5 consecutive peaks from large to small are 1, 171, 50, 1, 056, 4, 8.48, 942.43. Here this is the correction, 171.50, and 81, 8.3.39, and 684.35, and the calculated successive differences are 1.15, 0.02, 114.05, 129.04, and 129.04. Thus, based on known amino acid masses, they sequence the sequence of ASP, ASN, glutamine is the sequence. Automated programs are available that distinguish the ion types of a scan, remove as much background noise as possible, and calculate the most likely amino acid sequence. Protein identification does not require complete sequencing of all the peptides. Frequently, partial sequences of 2 or 3 peptides are sufficient for effective similarity researches of protein databases. A system called shotgun proteomics that circumvents cumbersome 2D separation of proteins in a gel uses liquid chromatography combined with MS, LC, MS for analyzing the proteins of a proteome. In this case, the entire mixture of proteins is in a sample is initially treated with a protease. Then the peptides are separated by LC, and the amino acid sequence of each peptide is determined by MS. Finally, the proteins are identified by database searches. Hundreds and in some cases thousands of proteins, including those not well resolved by a 2D page, have been recorded for proteomes and subproteins with this approach. Protein expression profiling is important for pinpointing changes during disease processes, cataloging differences between normal cells and cancer cells that can be used for diagnosis, and to tracking cellular responses to toxic agents. Both gel and non-gel methods have been developed for comparing the proteins of different samples. 2D differential in gel electrophoresis is very similar to 2D page. However, rather than separating proteins from different samples to individual gels and then comparing the maps of the separated proteins from 2 different samples are differently labeled and then separated on the same 2D polyacrylamide gel. Typically, proteins of one sample are labeled with the fluorescent dye CY3 and those of a second sample with CY5. The labeled samples are mixed and then run together in the same gel. This is figure 512, which shows protein identification by amino acid sequencing using ASI-MS of spot containing an unknown protein from a 2D polyacrylamide gel is excised and treated with trypsin to produce peptides. The tryptic peptides are separated according to their mass by charge and by Z ratios, and the amino acid sequence of a selected peptide is determined with a mass spectrometer image. The selected peptide is fragmented and ionized to generate a ladder of small peptides that differ by a single amino acid. Only the Y ion ladder, Y1, Y2, etc. is depicted here. The differences in the masses of the fragmented peptides correspond to the characteristic masses of the amino acids. The unknown protein is identified by search in a protein database with amino acid sequences from 2 or more peptides. This is the illustration of that. MW for pH to have 2-dimensional acrylamide gels, excise port, MS profile, trypsin treatment, mass, peptide mass determination, mass M by Z, peptide masses, amino acid sequence, computer search, protein database. Thus, this which overcomes the variability between separate gel runs. 2 dyes carry the same mass and charge, and therefore a protein label with CY3 migrates to the same position as the identical protein label with CY5. CY3 and CY5 protein patterns are visualized separately by fluorescence excitation. The images are compared and any differences are recorded. In addition, the ratio CY3 to CY5 fluorescence for each spot is determined to detect proteins that are either up or down regulated. Unknown proteins are identified by MS. The isotope coded affinity tag ICAT method combined with LC-MS is another way of comparative proteins from different sources. An ICAT reagent consists of an affinity tag biotin, a carbon chain mass-encoded linker that is labeled with either 8 hydrogen light form H or 8 deuterium heavy form D atoms and a chemical. This is a figure 5.13 of the 2D differentiate in gel electrophoresis method of quantitative analysis of protein expression. The proteins of two proteomes are labeled with the fluorescent dyes CY3 and CY5 respectively. The fluorescence labeled samples are combined and separated by 2D page. The gel is scanned for each fluorescent dye and the relative levels of the two dyes in each protein spot are recorded. The gel is stained with the protein dye and each spot with an unknown protein is excised and treated with trypsin. The peptides are separated by ESI, MS, and MS, and the amino acid sequences of some selected peptides are determined. The protein is identified by a search in the protein database for a likely match with amino acid sequences from 2 or more peptides. This is the illustration on that. Take a screenshot and should help you in your notes. And a group that covalently binds to an amino acid, usually cysteine. Deuterium is a stable isotope of hydrogen that is twice its mass, that is 2 daltons. Thus, the mass difference between a peptide that's labeled with a light and a heavy ICAT is 8 daltons. This difference is readily detected by MS. The proteins of one proteome are labeled with a light ICAT and those of another with a heavy ICAT. The samples are mixed, treated with trypsin, fractionated, and passed through an avidin column to capture biotin, which is present only on ICAT labeled peptides, both the light and heavy versions. The purified ICAT labeled peptides are separated by LC before the introduction into a mass spectrometer. The key feature of these technique is that the same ICAT labeled peptides from the two samples produce a pair of signals with a defined difference in mass due to the light and heavy versions of the labels. The relative amounts of each pair of light and heavy peptides reflect the relatively abundance of the source proteins in the original samples. Finally, with ESI, MS, and MS, the amino acid sequences of the peptides are determined, and the proteins that match the sequence can be identified. Hundreds of proteins from different samples can be compared in this way. Protein microarrays. Right? Conceptually, protein microarrays are similar to DNA microarrays. However, rather than arrays of oligonucleotides or genes, protein microarrays consist of a large number of proteins individually immobilized in known positions on the coated surface of a glass slide or silicon chip. The proteins arrayed on the surface can be antibodies specific for each protein in an organism, purified recombinant proteins, or short synthetic peptides. There are many ways of attaching a protein to a support surface. The major objective of any coupling system is maintenance of protein structure and function. Some systems bind the proteins to a chemical group that coats the surface of the support. With other protocols, recombinant proteins are prepared with a short amino acid sequence tag at the N or C terminus that binds to a recognition sequence on the support. In this case, all the protein molecules are uniformly oriented. In addition, instead of spotting proteins on a flat surface, some microarrays are engineered with tiny depressions, nanowells, that keep each protein moist and prevent mixing with an adjacent protein. The purpose of protein microarray analysis, for the most part, is to detect at large scale the molecules that a protein interacts with. These interacting molecules can be other proteins, nucleic acid sequences, or low molecular weight compounds. Protein populations from different samples can be compared, for example, in control versus treated samples, or normal versus diseased tissues. There are a number of methods for visualizing the interactions on protein microarrays. One common approach is to label the test samples directly with a fluorescent dye and then detect the labeled molecules and bind to the proteins of microarray with a laser scanner. A two-dye strategy, example CY3 or CY5, similar to that employed for DNA microarray analysis, can be used to compare proteins in two different samples and on a single array. Sandwich-style assays are also performed. Briefly, proteins in a sample are biotinylated and applied to the microarray. After the microarrays are washed to remove unbound proteins, streptavidin with a conjugated fluorescence dye is added and the sample proteins that bind to the proteins on the microarray. This is figure 5.14 of the ICAD method for quantitative analysis of proteins expression. A schematic representation of an ICAD reagent, and an ICAD reagent has either all hydrogen or deuterium atoms at the site X of a linker sequence. B ICAD protocol, proteins extract from two proteins, and the protein is labeled with light H hydrogen only ICAD reagent, and the other with heavy D deuterium only ICAD reagent. The samples are combined and treated with trypsin. The ICAD labeled peptides are captured by affinity chromatography using aviding, which binds to iodine and fractionated by LC. The ratio of the light to heavy H to the version of the same peptide is determined by AIIMS, which provides an estimate of the relative amounts of proteins in the two proteomes. The protein represented by a pair of heavy and light peptides is identified by amino acid sequencing with the SI, MS, and search in the protein database with the sequence for a likely match. This is the diagram. Take a screenshot and that should be able to help you. Now, and that's detected by laser scanning. In principle, the interpretation of the signals from a protein microarray is very similar to analysis of DNA microarrays. Broadly speaking, there are three types of protein macroarrays: analytic capture, reverse phase, and functional. Analytical macroarrays are used for a protein profiling, that is the detection and quantification of protein space present in a sample. It consists of either protein samples applied to immobilize antibodies, antibody samples applied to immobilize proteins. Antibody microarrays are often probed with proteins from biological sources such as serum or plasma, or proteins that are secreted from cells in the culture to determine disease-specific proteins profiles. For example, antibody microarrays that specifically detect cytokines have been formulated. Cytokines, if these are a large number, are small secreted proteins, signaling proteins that mediate and regulate immune and inflammatory responses, cell death, apoptosis, cell growth, blood vessel formation, and differentiation in humans and other animals. Cytokine antibody microarrays are used to examine cytokines in both normal and disease states and from a variety of sources after various treatments. A sandwich immunoassay is often used to detect cytokines that bind to immobilized antibodies. After the microarray is treated with a sample containing cytokines, biotinylated cytokine antibodies are added. And a biotinylated cytokine antibody will bind to a cytokine that is bound to an immobilized antibody. Streptavidin with an attached fluorescent dye is added next. Then the signals are detected with a laser scanner and the data are analyzed. In one study, plasma samples from individuals with Alzheimer's disease and those from individuals with no dementia were applied to a microarray made up of antibodies against 120 cytokines. 18 cytokines were found to be associated with Alzheimer's disease. The levels of these were, seven of these were higher with 11 were lower in individuals with Alzheimer's disease than in the subjects without dementia. The Alzheimer's disease-associated cytokines adversely affect blood cell formulation, immune response, apoptosis, and nerve cell signaling. The Alzheimer's disease cytokine pattern was also present in individuals with mild cognitive impairment. Currently, there is no definitive method for either predicting Alzheimer's disease or determining whether a patient has the disease. Possibly, the Alzheimer's disease-specific cytokine signature may provide the basis for a diagnostic test. for this degenerative disease that afflicts more than 4 million people in the United States alone. Now this is figure 516 is the detection of cytokines with the cytokine antibody microarray, and the other is the protein microarray detection methods, direct labeling, and sandwich style assay. Another type of analytic microarray proteins, antigens, are attached to a solid support and then probed with antibodies, mostly in serum samples. The purpose of these studies is to discover whether the production of antibodies against specific proteins correlates with particular diseases or biological processes. By way of illustration, a microarray with about 5,000 different human proteins was created and used to determine serum from ovarian cancer patients has a distinctive set of antibodies in comparison to the antibody population of healthy individuals. The initial results revealed 94 proteins that were specifically recognized by antibodies in the sera from the ovarian cancer patients. With further testing, 3 proteins were consistently found to be specific for ovarian cancer. The earlier ovarian cancer is diagnosed, the better the chances of survival. Unfortunately, ovarian cancer is currently identified at a late stage. The ovarian cancer specific protein may help in the detection of the disease. Antibody microarrays have also been used to determine whether particular post-translational protein modifications such as phosphorylation, tyrosine, or glycosylation are associated with specific diseases. To screen the proteins that contain phosphotyrosine proteins are first captured by primary antibodies immobilized on a microarray, and then the microarray is flooded with biotinylated anti-phosphotyrosine antibody. Next, streptavidin conjugated with the fluorescent dye is added, and the protein spot with fluorescence is detected in a similar manner. Glycosol groups, glycans present on proteins can be visualized by adding a biotinylated lectin to proteins that are bound to immobilized antibodies on an array. Lectins are plant glycoproteins that bind to specific carbohydrate moieties on the surface of proteins or cell membranes. And many different lectins with affinity for different glycans are available. The use of lectins can facilitate the direction of specific protein glycosylation patterns. The utility of antibody microarrays has been enhanced by the expansion of libraries that produce highly specific antibodies. For example, clones making antibodies against more than 1,800 human proteins have been isolated, characterized, and validated. The long-term objective of this project is to have an available and one antibody for each protein from every coding sequence in the human genome. That is a total of about 22,000 antibodies. With the reverse phase microarray, a multi-protein sample, for example, from a cell lysate or tissue specimen, is immobilized in a single spot on a support. Several such multi-protein samples are spotted on the microarray, which is then probed with a single target molecule. This format contrasts with analytic and functional microarrays in which immobilized spots containing single proteins are probed with multiple targets. Hence the term reverse is used. The advantage of reverse phase microarray is that a large number of samples can be compared at one time. With a reverse phased microarray, the presence of specific proteins in multiple complex samples can be readily determined. Function and protein microarrays feature large sets of individual proteins that are used predominantly to determine interactions with other proteins or lower molecular weight compounds such as lipids, drugs, and metabolites. Enzymes such as oxidases, kinases, proteases, and glycosidases with novel activities that are useful for biotechnology and medicine are likely to be discovered using functional microarrays. Ideally, a functional protein microarray should consist of all possible proteins of a proteome under study. To obtain comprehensive representation of a proteome, a library containing all of the protein coding sequences is first constructed. The term ORF, which stands for an open reading frame, is used to represent any protein coding sequence. And a library of clone protein encoding ORFs have been dubbed an ORFiome, large-scale expression of the library is followed by clarification of each of the proteins, which is subsequently arrayed on a solid support. Detection. The figure 517 shows detection of post-translational modifications with antibody macroarrays. On the other hand, full-length cDNA libraries are the primary source of coding sequences of a eukaryotic protein. We will be ending this lecture here in a couple of seconds from now. The rest of the portions will come in on the next lecture. So take notes, take screenshots, be well, start preparing well for your exams. Best wishes. Thank you.